My name's Julien. I'm a PGY5 for those who don't know me. This morning I'm very excited to tell you about my research project, which is optical stimulation of the cochlea. First, I wanted to answer the question, why would you even want to shine light on the cochlea? Because it's a hearing organ. And for me, that story goes back to graduate school. For grad school, I was in New York in the lab of Jim Hudspeth. Jim is an amazing scientist. He's done tons of experiments and brought the field forward quite a bit. He was very driven by questions and would always go for the big questions and then make the science work. And so some of us in his lab had kind of this motto that we felt was the lab's motto, which was making the impossible nearly difficult. So I'll start off by giving you a little bit of a flavor of what we did in Jim's lab and then what we did here. So we all know that the cochlea converts pressure into our perception of sound, and it does so using the organ of Corti, which you can see here. These hair cells with a tectoral membrane on top are stimulated by the pressure waves in the cochlea. If you zoom in, you see hair bundles at the top of hair cells, which give them their name. And these hair bundles allow ions to enter into the mechanically gated channels at the top of each hair cell. So here, I'm zooming in on just two stereocilia. You see this little filamentous protein called a tip link. And when these bundles are pushed over towards their tall edge, the tip link stretches, the channel opens, and you get a current entering the mechanotransduction channel. That's how you convert pressure into electricity. But we know that obviously current enters, most of the current's carried by potassium, but there was a lot that we did not know. For example, calcium seems to have some kind of role, but we just didn't quite know what. There was some evidence at the time from labs, including Tony's and many others, that calcium could have been binding to the channel or elsewhere in the hair bundle. And the experiment we were trying to do was to understand what's the role of calcium inside of the hair bundle. To do this, I was tasked with trying to release calcium in a controlled fashion inside of a single cell. So we try to do this using what's called uncaging. Uncaging is where you protect your molecule of interest with a cage, and then at the time of your choosing, you zap the molecule with UV light, crack the cage open, and you release your molecule of interest. So in our case, we did this with calcium to see what the effect of calcium would be on the hair bundle. I started working on this, got some data, and one day I thought, I should probably do a control where I just shine a light on the hair cell without the caged compound just to make sure nothing happens. And much to my dismay, what I found was that if you just shine light on a hair cell, turns out the hair bundle moves. All the bundles were moving forward towards their tall edge. This made no sense. How is a hearing organ responding to sound, and why is it even moving? First of all. Second of all, how am I supposed to do my experiments now that my negative control is showing motion? And so I was a little bit panicked, to say the least. This was about a year into my graduate work. And I want to show you a video of what I saw at that time, because it was a little bit, for me, it was impressive. Just so you get a feel of what it looks like. Before the video, just to orient you, this is a frog hair bundle. This is a scanning electron micrograph. You see the tall edge here, the short edge here. If you push this thing towards its tall edge, you open the channels. On the right is an epithelium from the frog sacculus. And you see a lot of these hair bundles. Here's one, here's another, here's another. All these white dots are hair bundles viewed from the top. If we zoom in to an image like the one on the right, you'll see something like this. Here I'm showing you two hair bundles. There's one at the top of the screen on the right side and one on the bottom. They're facing opposite directions. So there's a little bulb here called the kinociliary bulb. It's facing to the left. That one's facing to the right. So these two bundles are kind of looking away from each other. And when I start the video, the UV laser will pulse repeatedly. So I was expecting to see nothing, and this is what I saw. All the bundles in my field were moving towards their tall edge in sync with the light. It was wild for me, to say the least. I thought there was an issue with the camera. I thought maybe the whole epithelium was moving. Honestly, I didn't know what to think. But I did a bunch of experiments during those 2 weeks. One of them was to get the hair cell out of the epithelium, get it to lay on its side, and get an idea of, is the motion happening really only at the bundle, or is the cell body moving as well? So here's one of those experiments where this cell has been taken out of the epithelium. It's lying on its side. So the tall edge is here, the short edge is there. This is with a high-speed camera. And here I'll start the video, and again, it'll pulse. So I thought this was really cool, also really annoying, but it's clearly just the hair bundle moving towards the tall edge. So after getting over my initial disappointment, I actually took this on as my main project, trying to figure out why is this even happening. I would say these 2 weeks, the initial 2 weeks period after we found this were kind of almost a sub-manic period for me, where every day we had a new idea. I would try it in the lab, get more information, and it was kind of the happiest I've ever been in life. And I just want to give you a flavor of a couple of the experiments that we did. So off the bat, one of the things we wanted to try was to break the tip links. So these are the links connecting each stereocilium to its taller neighbor. Turns out that if you break the tip links, the motion I showed you goes away entirely. That all of a sudden made it more interesting because the tip links are connected to the channels. So then I thought, well, if that's the case, what if I record from the cell using a patch electrode or a sharp electrode? I recorded from the cell, and lo and behold, in response to the light, those channels opened too, and you could block it with a channel blocker. So now we were in business, and it felt like it was worthwhile going after this and trying to figure out how it worked. Some of the experiments we did were, for example, this one, where I would try to shine the light on only a smaller region of the hair cell, like the hair bundle or below it, or a combination of both, to try to figure out where was the light-sensitive element. I did this with a scope where basically I miniaturized a projector, like the one in this room, so that I could shine it onto a single hair cell. Or another experiment was looking at the effect, meaning the motion that we saw as a function of wavelength, to get a better idea what the absorber was. There were many more experiments, most of which didn't work, but the ones that did work made it into that paper, so I'll show you a link at the end if you're interested. What we ended up finding was that there are intrinsic absorbers inside of hair cells. They absorb UV light, they then convert it into heat, and the heat ends up changing the mechanotransduction apparatus and opening the channel, the mechanically gated channel. So that's kind of how I got interested in shining light on hair cells. It was a little bit of an accident. But during the course of this work, of course, I did a literature search to figure out if anyone else was trying to do this type of work. And I ended up finding that, yes, indeed, people were trying to shine light on the cochlea, but infrared light. And before I tell you why people are trying to shine infrared light on the cochlea, let me just show you that it does work in terms of stimulating the cochlea. This is data from Klaus-Peter Richter's group in 2008, where this was a rodent cochlea, and they shown different intensities of light. So as you shine more and more infrared light, and they recorded an auditory brainstem response, which is an auditory evoked response. So right off the bat, you can see, okay, if you shine IR light on the cochlea, you can get a response. The reason why they were even trying to do this was basically to improve their current cochlear implant. For those of us in the business, sensory neural hearing loss comes around mostly due to defects in transduction or loss of hair cells. Our best treatment for this is to bypass hair cells using a cochlear implant and stimulate the spiral ganglion neurons directly. This works really well, and hundreds of thousands of people have had their hearing restored thanks to cochlear implants. But the downside is that the implant uses electricity, which spreads within the cochlea, and it's kind of like playing the piano with your forearm, not a great way to carry a melody. And so the idea with light, and in particular infrared light in this case, was that maybe the light can be more tightly focused, and maybe you can stimulate the cochlea in a more focused fashion, allowing better noise in sound in noise recognition and music enjoyment for cochlear implant recipients. The downside is that nobody knows how IR light works. We don't know if it stimulates the neurons directly, or the hair cells, or even both. The mechanism is just not known. There are many candidate mechanisms that have been published, but nobody to date knows the actual mechanism of stimulation. I think that's pretty crucial if we're going to use this in a translational manner. Despite not knowing the mechanism, there is a clinical trial ongoing, primarily at Northwestern and Miami. They are asking patients who undergo translab vestibular schwannoma resection for 30 minutes extra under anesthesia so they can use infrared light to stimulate the cochlea. For me, coming out of grad school and starting residency, this wasn't satisfying. I really wanted an answer in terms of a mechanism to how IR light works. And I wanted to answer the question of, is this something I could potentially spend the rest of my life on, making a device to stimulate the cochlea and improving the next generation of cochlear implant? So I sought to answer these questions, and that's what I hope to answer for you today. How does infrared light stimulate the cochlea? And can it underlie the next generation of cochlear implants? So hopefully by the end of today, we'll have these questions answered. To do this, I joined Tony Ricci's lab here at Stanford. Tony is a well-known electrophysiologist, and it has been an absolute joy working with him. I don't think this project could have been done without Tony. And so I approached him early on. We talked about the project and different ways that we could use technologies in the lab to get it done. And it's been really an awesome journey, as I hope you'll see. When I realized that's the only picture I have of Tony and I by ourselves. So the way that we wanted to approach the project was to develop a system where we could record from a mouse and stimulate with both light and sound in the very same mouse. That way we would have sound as kind of a backup and a control. So many of us are familiar with the auditory brainstem response. This is an evoked response to sound. So you can put electrodes on babies, or you can put electrodes in mice, play a click or a tone, and you get this response, which it's pretty stereotyped. It's got multiple peaks. Each peak represents a different nucleus along the auditory pathway, and we're supposed to memorize them for boards. The ABR, in a way, is similar to the EKG. The EKG just happens to record the heart activity. And we're all familiar with the EKG. But just like the EKG, if you want to change what you pick up, meaning, let's say you're worried about the back wall of the heart, you can put the electrodes on the back of the patient to get a better recording of that area. You can do that with the ABR too. In my case, I wanted to pick up more cochlear information from the hair cells and from the spiral ganglion neurons. So I added an electrode to the round window, which is right on the cochlea. This gives me a record that's similar to the auditory brainstem response, but it's bigger. You see it's tenfold bigger. This initial peak is often called the compound action potential. It's the same peak as this first peak here in the ABR, and it's the sum of the thousands of spiral ganglion neurons in the cochlea firing at the same time. The subsequent peaks are less emphasized because it's further from the brain. So I chose to record both of those at the same time. Here's a typical mouse experiment. I can show this safely in this room. So this is a left neck, and I just want to show you the electrode configuration for the experiments. So this is the mandible of the mouse. That's the masseter. The snout would be somewhere up there. Here's the pinna, or what's left of the pinna. This is one of the ABR electrodes piercing this SCM and going through the pinna. Here we have the bulla, which is the equivalent of the mastoid in a mouse. I opened it up, but I preserved the tympanic membrane and all the bones of hearing so that I could get access to the cochlea, but the mouse would still be able to hear. There's another electrode here, a really fine one, going into the round window, so I can simultaneously do round window and ABR recordings. And then the big orange fiber here is an optical fiber carrying the infrared light directly on the cochlea. So this mouse can have an ABR, a round window recording. It can be stimulated with light and stimulated with sound using this preparation. So in terms of the recording that we did with the round window, I wanted to be able to pull out not just the neural activity, but also hair cell activity, because the core question here was going to be, is IR stimulating neurons or hair cells? So we tried to pull out the hair cell signal, and let me show you how we did that. This is the trace I showed you earlier with the round window recording, or the compound action potential. And this is what it would typically look like if you record from someone, let's say, in response to a click. But in fact, there's data buried within this trace. When you play a click with a speaker, the speaker cone can either move towards you or away from you. It's called a compression or a rarefaction. Typically, in hearing tests, we average those two. You do both stimuli and you average them. The reason for that is that you want to average out the hair cell component of the response and only have the neural response. So let me walk you through this. Let's say the speaker cone moves towards you. You get a compression. The stapes is going to go into the cochlea, and the hair cells will be moved in one direction. The hair bundles will be moved in one direction. Now you do the opposite. The speaker cone moves away from you. The stapes moves out of the cochlea. The bundles move the other way. At rest, the bundles have some fraction of their channels open. So this means that hair cells with these two stimuli will respond in opposite directions. In one case, they'll be depolarized. In the other, they'll be hyperpolarized. And we see that in this trace because there's this high frequency kind of noise superimposed on top of the neural response on top. And that high frequency noise, really, it's a hair cell signal, can be pulled out. To pull it out, all you have to do is keep each phase of your recording, subtract out the nerve portion from here. So for example, if I have the orange trace here and subtract the black trace from the orange, I'll get this. This gives me the hair cell component of the response. And this gives me the hair cell component of the response to the opposite phase. And so usually we average these out. If you add this to that, you get 0, you get a flat line, and the hair cell signal disappears because most people only want to look at the neural signal. In our case, we kept all the data so that we could pull out this hair cell response. So in summary, the top one can give us neural activity, and the bottom one can give us the hair cell response. So I'll show you that in the traces in a second. So here's a typical experiment that we did. This is in a wild-type normal hearing mouse, and these are sound stimuli. So first on the left, this mouse in response to a click has a normal ABR. It also has a normal round window response. You can see a nice compound action potential. And if we pull out the hair cell signal, like I just showed you before, you see a nice big hair cell component of the response. So this is a total normal mouse. Now instead of sound, you can use light. And this is what you get. In response to infrared light, you get a normal ABR, and you also get a compound action potential. Now, both of these look a little bit different from sound-evoked stimulation because there's this kind of sharp peak at the beginning that you see in both the ABR and the compound action potential. We'll come back to that in a second, but the basic point is you can stimulate the cochlea with IR light. So now that we have the setup, that I've explained the setup, let me show you the first manipulation that we did. Like I said, we wanted to know whether hair cells or neurons are the target. So the first thing that I tried was to separate the two. I used a mutant called the VGLU3 mutant, vesicular glutamate transporter 3 knockout. This transporter fills the vesicles at the bottom of hair cells with glutamate. So when you knock it out, the vesicles are empty, and the hair cell basically cannot talk to the neurons. Even if you stimulate the hair cells, the neurons will never hear from them because they're totally disconnected. So this is a great way to test whether you actually need hair cell input onto the neurons for IR stimulation. So I'll show you those results. This is a wild-type littermate. This is actually the slide I showed you earlier. And here's the VGLU3 mutant. In response to sound, there's no ABR. That makes sense because the hair cells are unable to talk to the neurons. There's no round window response. That also makes sense. But if you look at the hair cell component of the response, it's still there. That also makes sense because the VGLU3 mice have hair cells. The hair cells work. They just can't talk to the neurons. If we now look at what happens with infrared light, you get no ABR in response to infrared light. That also makes sense because the hair cells can't talk to the neurons. And when you look at the round window response, it's missing the big action potential, which would be here, the compound action potential. Interestingly, in the IR case, you still have this fast early spike here, which we also noticed in the wild type. And I wanted to make the point earlier that in the case of the laser, what you don't see here is a microphonic. There's no third column in the laser, right? That's because with the laser, you can't use inverse phase to pull out the hair cell signal, right? With sound, there's compression, rarefaction. You can use that to pull it out, but laser's either on or off, and so we can't pull that out. That means that the hair cell signal is added and buried into this signal here. So in the laser, the round window response contains both. They're inseparable. And interestingly, in the VGLUT3 mutant, we're left over with this fast spike. So naturally, we wondered, is what is left over here, is that the hair cell response? Is that what hair cells do in response to IR light? And to make it more clear why we wondered that, I'm just going to blow up those two traces on the right and overlay them one on top of the other. You can see that the VGLUT3 in red doesn't have that action potential. The black action potential is only in the wild type, but that fast early response is in fact present in both, and it's almost identical. So to figure out whether that was the hair cell signature, if you will, to IR stimulation, the next step was to kill the hair cells and see if that was still there. To kill the hair cells, I used a mutant called the POW4F3 DTR mutant. It's a mouthful, but basically this mouse expresses the human diphtheria toxin receptor only in hair cells. So the mice grow up normally, they hear just fine, but that at a time of your choosing, you inject them with the toxin, and then the hair cells start to die. So it's a great way to kill hair cells in a controlled fashion. So here's the data from that mutant. Up top is another control. This is a wild-type control that got the toxin, but obviously because it didn't have the receptor, it doesn't care about the toxin. And on the bottom is a diphtheria toxin mouse. No ABR, because there's no hair cells, no round window response, and the hair cell component of the response is also pretty much gone. There is a tiny little bit of signal, and actually we didn't kill all the hair cells, and that's probably why we see that. In terms of the laser response, that peak that we saw there in the VGLUT3 is gone. Just for comparison, this was the VGLUT3 data. So you see that that hair cell component is now gone. So I think these two experiments on their own already answer the first question that I posed to you early on. It turns out that infrared cochlear stimulation does require hair cells. This was, I would say, not maybe the best news, but we wanted to know how exactly it worked. We still wanted to try to get a mechanism to see whether this could be useful for something else. And there were many potential mechanisms that we thought about. It could be that infrared light targets the hair bundle somehow. There was some evidence that it might target mitochondria, that it could change the capacitance of the cell membrane, or perhaps any other options. So the first thing that we targeted was the hair bundle. Is infrared light somehow stimulating the hair bundle? To answer this question, we chose to basically take the battery out of the cochlea. Let me explain what I mean. In the cochlea, in the scala media, there's something called the endocochlear potential. This is a positive 80 millivolt potential or battery that pushes ions into hair cells anytime the channels are open. So if you take this potential out, it's like taking the battery out of your toy. Even if the hair bundles, if the channels are open, there will not be a driving force entering into the hair cells. So it's a great way to kind of take the legs out of the hair bundle, but keep the rest of the hair cell intact. If the light was going to stimulate the mitochondria or the capacitance of the cell membrane, it would still be able to do that. To take out the battery, so to speak, we used furosemide. As we know from boards, furosemide targets the stria vascularis, which is the tissue that generates the endocochlear potential. And in this nice figure from Elliott Strimbu, you can see that if you plot the endocochlear potential as a function of time, each color here is a different mouse. They start at positive 80 millivolts, and then at time 0, when you inject the furosemide intravenously, the potential drops down to about minus 20. It stays there for maybe 10 minutes, and then it starts to recover. So the idea is, let's use those 10 minutes when there's no endocochlear potential, no driving force, and see if you can still stimulate the hair cells with IR. So that's what we did. Here's our version of that data. On the y-axis, I've got time after furosemide injection. So negative time is before the injection. So at t minus 20, the threshold of hearing was 30 decibels. I'm plotting the threshold here on the x-axis. So this is a normal hearing threshold for a mouse. The threshold stayed the same 10 minutes later, and then at time equals 0, I injected the furosemide, and boom, the mouse goes deaf. 100 decibels doesn't give me a response. Then after maybe 10 minutes, it starts to recover and the hearing gradually gets better. Another way to look at this is to just show you the ABR trace for the 90 decibel stimulus. It's a little bit easier to see. You see a nice ABR, it continues to be nice, and then after the furosemide, the mouse is deaf, it stays deaf, and then it starts to recover. Between these two time points, when we know for sure the mouse is deaf, that's when we want to hit it with IR, because we know for sure there's no endocochlear potential during that time. And here's what we saw. The right column is the infrared stimulation. You see that you can get an infrared evoked ABR just fine before the furosemide, but when the mouse is deaf, you can't elicit an ABR. So this made us think, okay, probably the hair bundle is involved in some way. The battery of the endocochlear potential seems to be necessary for this response. The hair bundle is a mechanically sensitive organelle. Its day job is to sense displacement and convert that into electricity. So the next logical step was to look for a mechanical displacement in the cochlea in response to infrared light. For this, we traveled down to LA at USC and worked with Dr. Ogilai and Yuki, who's Dr. Ogilai's postdoc, using optical coherence tomography. This was the one night when we went out to the Baked Potato for some drinks, but otherwise we were pretty much in the lab lasering mouse cochleas day and night. The technology they have at USC is a particularly good version of optical coherence tomography, which allows you to detect motion within living tissue. I'll just give you a 2-second primer on optical coherence tomography. Basically, the idea is that you have a sample and you're trying to detect the motion of your sample. You do that by having an imaging laser that you shine on your sample. But on the way to the sample, there's a mirror that reflects the light to a reference mirror, and then the reference mirror will reflect it back to the detector. We'll call this the reference beam in green. That beam is constant in time. But your sample, if it's moving, let's say it's moving up and down because you're looking at a membrane in the cochlea, its reflection will also be kind of moving up and down, so to speak. And when it gets reflected into the detector, that light will interfere with the reference beam, and that interference pattern will tell you about the motion of your sample. So the idea is, let's use this and then shine the light in different parts of the cochlea, like the organ of Corti, the tectorial membrane, or different parts, and see if there's any motion and see if the motion is different in different spots. So that's what we did. In real life, it doesn't look nearly as beautiful as that cartoon. This is a typical optical coherence tomography. The image, the SM shows you scala media. So just to orient you, this is otic capsule bone here. This is Reisner's membrane, which is a separation between scala media and scala vestibuli. We have the organ of Corti here. You have to be an aficionado to be able to tell. This is tectorial membrane. And so it's kind of analogous to this image here. So our first step was, okay, let's put the laser on the bone and see if the bone is moving at all in response to infrared light. So that's what we did. And here I'm plotting displacement of the target tissue on the y-axis as a function of time. The gray bar shows you when the infrared laser is on. You see that you can shine infrared light, the bone doesn't move, it doesn't care. So next we decided to shine the light on something more flexible, like a little membrane inside the cochlea. We chose Reisner's membrane, which is kind of a passive boundary. There's nothing active about it. And interestingly, after the infrared light, we saw a gentle wave of motion here on Reisner's membrane. Note taken. Then we looked at the organ of Corti, which is down here, in a dead animal. Similarly, there's a little gentle wave of motion after the infrared light. But when you look at that same spot in a live animal, all of a sudden things get a little bit more interesting. The motion is bigger, the amplitude of that wave is bigger, and it's followed by some little wavelets. And if you specifically put the beam where outer hair cells live, here shown with the red X, it's even bigger and the wavelets are more prominent. And I'll show you another example on the next slide too. So this made us wonder, is it the case that IR light is generating a little bit of motion within the cochlea, and that maybe the organ of Corti, which is a mechanically sensitive organelle, is picking that up and amplifying it? In fact, we know outer hair cells, their number one job is to amplify sound, while inner hair cells detect sound. Another experiment we wanted to do right off the bat is to, in addition to detecting the displacement of the organ of Corti, couple that with the electrophysiological response and measure both at the same time. And that's what I'll show you here. At the top is another example of infrared evoked displacement. This example is a little nicer. You see the big wave and then followed by this ringing after it. At the same time, we recorded from the round window in this mouse. And you see the action potential, which I've been showing you earlier in the talk, and also that fast early spike. This trace is kind of blown up in time compared to the ones from earlier in the talk. Interestingly, those two look pretty similar, especially in the beginning, and if you overlay them, that becomes even more obvious. Here I'm blowing up the red shaded region underneath. You can see that in black, you've got the mechanical displacement, and just a few microseconds later, you've got the electrical potential. So something that, generates a potential a few microseconds after motion is very much a hallmark of the hair bundle or of mechanotransduction. It is extremely quick, quicker than our sense of vision, and this is very much in line with recording from hair cells or from a mechanically stimulated basilar membrane. So I mentioned before that we thought perhaps the outer hair cells were picking up that little wave of motion that I showed you and amplifying it. But the data I showed you doesn't really is not enough to convincingly say that that is the case. We wanted to probe that a little bit further and see what is the role of outer hair cells. To do that, we used a mutant called the tecta mutant. In this mutant, the tectorial membrane, which normally sits on top of the hair cells, is kind of lifted up, and it doesn't really touch the outer hair cells. Here's a comparison. In the wild-type mouse, or human even, the tectorial membrane sits right on top. That means that in this mouse, when the gentle wave of motion goes through, we would expect the outer hair cells to not get stimulated. And so we were curious, are we still going to see that amplification? And here's what we saw. You already know that in wild type there's a big motion ringing, and this is the electrical response. And in the tecta mutant, we only had this passive wave, even when we recorded from this spot here, and it was electrically quiet. In other words, if the outer hair cells are not stimulated, you don't get everything that comes afterwards. You can compare this to the same experiment in the VGLUT3 mutant. Remember, this is the one from earlier in the talk where the synapse is silent. This mutant has totally intact hair cells and an intact tectorial membrane that sits on top of them. So it's able to amplify just fine. In fact, we found that these mice amplified even more than wild-type mice for reasons we can go into later. And it doesn't have the action potential because the synapse is silent, but it does have this fast electrical response. So this shows us that if you have functioning outer hair cells, you do pick up that signal and you amplify it. Then of course, you need a working synapse to send that signal onto the brain. So in conclusion, what I think I've shown you so far is hair cells are required for infrared stimulation of the cochlea. The infrared irradiation generates motion within the cochlea. That motion is amplified by the outer hair cells and then picked up by inner hair cells. If this framework is correct, we should be able to break this whole system down by just getting rid of the mechanotransduction channel in the hair bundle. That's the channel up top here that allows current to enter through the hair bundle. That's because this channel is necessary both for outer hair cells to do their work. They have to be stimulated to be able to amplify the sound, and it's necessary for inner hair cells to detect the sound. So we decided to do this as a confirmatory experiment. What we used was a mouse called the TMC1 knockout. It's missing the protein for the mechanotransduction channel. So this mouse is deaf and the bundles cannot transduce. And what we saw in this mouse was basically all across the board silence. There was no ABR, no compound action potential, no hair cell component of the response to either sound or laser light, kind of confirming the mechanism that we put forward. So these were the two questions I set forth earlier in the talk. I think that I've gone through the mechanism of how light stimulates the cochlea, infrared light stimulates the cochlea pretty well. One thing you might wonder is, well, why is IR light even generating that little wave of motion? Why does light cause that? That's because IR light's actually absorbed by water very, very strongly. And the pulse of light that I use is 100 microseconds long. It's very, very short. And so we're operating in a system, in a framework called thermal confinement. Thermal confinement is when you deposit heat within an area in a time that is shorter than it would take the heat to diffuse away. So we know that if you heat something, the heat diffuses away, right? But if you deposit it within a short amount of time, the heat is building up there, and then when you turn off the stimulus, all of a sudden this area is way hotter than the surrounding and it expands all at once. So that's how basically a pressure wave is generated within the cochlea. It's a tiny little pressure wave, but it's just enough to stimulate the hair cells. Can infrared light underlie the next generation of cochlear implants? Well, I think that I've convinced at least myself that I don't think so. It seems to really require hair cells to be functional. When you take out any component of the hair cells, it doesn't work. And so, unfortunately, I don't think this will be a translational application for us. I do think it's potentially still useful because, for example, this gives us a way to stimulate the cochlea without even touching it. That could be quite useful in the lab if you're taking the cochlea out of an animal and you want to be able to stimulate it without damaging it or probing it or breaking the bone from the outside, but probably not going to be seeing it in patients. Our paper on this actually just came out about a week and a half ago. So if you're interested in seeing more experiments and some stats, that's the paper that just came out. This was the paper down here from a graduate school where we have a lot of other experiments on the frog UV stimulation. And then that's a nice paper that's a review of infrared stimulation and other work that's been done so far. So you might be asking, what's next? What can we do next? And it turns out that in parallel with the infrared stimulation of the cochlea, other efforts have been going on by other labs with optogenetic stimulation of the cochlea. Optogenetics is when you take an intrinsically light-sensitive channel, like channel rhodopsin, and then you insert it into neurons using a virus. So you can turn neurons that were not light-sensitive into light-sensitive neurons just by transfection. So that could allow you, for example, let's say that all these neurons now carry an opsin. That could allow you to make a cochlear implant where you shine, let's say, green light, and the light can be focused, just like IR light, and you could potentially have better frequency resolution. It turns out that we've known for some time that these neurons don't all come in just one flavor. There's at least 3 or 4 flavors, and some of those flavors are different thresholds. There are neurons that respond to high threshold sound, medium, and low threshold sound. And recently, over the past few years, people have found that there's actually different genetics in each of these neurons, and they've been able to genetically separate them. People are also working on different promoters for each of these subtypes. And so I could envision a world in which each of the 3 subtypes could get a different opsin, and not only can you stimulate in a more with better frequency resolution, but you can also triple the dynamic range of the cochlear implants where based on the loudness, you fire a certain color. And so this could potentially drastically improve implants in 2 different modalities for patients. Another interest that I am thinking about of working in myself is diagnostics for sudden sensory neural hearing loss. I think many of us in the clinic know that when a patient walks in with sudden sensory neural hearing loss, all you do is you have an audiogram and you either do steroids or not, and there's not much more that we can do. It would be nice to have some diagnostic test to tell you, is, for example, the battery out of the cochlea, or is it something else that's going on, and can you potentially treat it with some other medication than just steroids?
What?
To conclude the talk, I've already thanked Jim and Tony, who are my 2 closest scientific mentors, but I wanted to say another thank you to Dr. Sung and the residency program and the faculty. As many of you might know, when I matched for residency, my mom was diagnosed with metastatic cancer, and she passed away when I was a PGY2. That was a really hard time for me. But the program was extremely supportive. I took time off, and my co-residents and Dr. Sung, everyone was extremely supportive at that time, and I was able to get through that really hard time thanks to that. My mom was also a scientist herself, and that's her on the left there, and I wanted to dedicate kind of this talk to her because I feel like my passion for this kind of mechanistic science came from her. And finally, Rebecca, my wife in the middle, has been basically supporting me through all of this. When I come home late from lab or late from the OR, she's always there. And thank you to Rebecca. Thanks, everyone, and I'll take any questions.